things about the eye that you all need to know. First of all, the pupillary muscles, we may have mentioned this. Remember so what are the extrinsic muscles of the eye? Okay. So the rectus muscles, the four rectus muscles, and the two obliques. Okay, so those are extrinsic, which means what? On the outside of the eyeball. And then we have intrinsic muscles, such as these two, pupillary constrictor and pupillary dilators. So when one of these sets of muscles, and these are smooth muscles, so we don't have control over them. Huh? Can y'all hear? Pupillary constrictor and pupillary dilator muscles. And they control the diameter of the pupil. And what I wanted you all to notice is the direction of the muscle fibers. What? No, if you look at the picture, I don't know if you all have one close to you. But you can see the muscles, how they're arranged. So the pupillary constrictors muscle is circular. It's a sphincter. You all heard of sphincters? So sphincters are circular muscles. And what do muscles do? They contract. So if a circular muscle contracts, what does that do to the opening? It makes it smaller. So you can see then that if you stimulate a sphincter muscle, the pupillary constrictor, that causes the pupil to constrict, become smaller, allowing less light to come in. So you can see that happening on that side. And if I ask you about the parasympathetic division and what it stimulates, first of all, you know the parasympathetic division has what effect on the pupil? It causes constriction, right? So, but these are the muscles of the iris. So when the constrictor muscles of the iris contract, that closes or constricts the pupil. And the other muscles are radial muscles. They kind of extend around outwardly, not circular muscles. So if you look at those and where they're attached to the circular muscles. So when they contract and they shorten, they pull the sphincter muscles in order to open the pupil or dilate the pupil. Okay, so you have the pupillary constrictors, which are sphincters, circular muscles, and then the pupillary dilator muscles are radial, and they cause the pupils to dilate. Okay, so on the test I may ask you which division stimulates the radial muscles of the iris and which one is stimulated by the parasympathetic division. Okay, and it tells you underneath which one does what. So you all already know the sympathetic division dilates and the parasympathetic division constricts. Okay, so that's not the only thing that controls the pupils. Also, the amount of light you are exposed to will determine whether the pupils dilate or constrict. And why is that necessary to have that backup in case you're out in bright light or in dim light? Why is it necessary for the pupils to be able to change depending on those two environments? Well, not focus. So the pupil doesn't really. Right. So you don't want your eyes to get really the retina. The retina can't stand too much light. So in those situations where you're in very bright light, the pupils constrict to decrease the amount of light getting into the retina. And then if you're in darkness, you need more light, so the pupils will dilate in order to let a little bit more light come in. The retina, again, is where we have our photoreceptors, and the photoreceptors are rods and cones. So the rods are activated in dim light, and they allow us to see shades of gray. Cones, on the other hand, function in bright light, and they provide color vision. And cones are densely clustered in the fovea. What is the fovea? So it's that little part in the center of the retina. And what's important about it? Right. Provides high visual acuity, sharp vision. Now, so this is an interesting picture you may not have thought about when light comes into the eye and hits the retina. So this is a picture of the full retina. So this is the front of the retina. So light goes through this part of the retina, but it has to go through all of these other cells that we'll mention in a second in order to make it back there to the pigmented part of the retina, which is where the rods and cones are activated. So the pigmented layer is the very posterior part of the retina, and they absorb the light energy, and that activates the rods and the cones. So you all see the rods and the cones? What's the difference? Well, in the picture, the colors are what? What's the color of the rods? Yeah, so they look red up there on the screen, but they're kind of brownish. So they're the ones in the back, and then between those, a few of the cones are there as well. So the cones have a cone-shaped surface, and the rods have the rod shape. But the importance is there are functional differences. Okay? And now, so once those rods and cones are activated, just like any other receptor, they have to transmit the signal to the correct part of the brain, which is what? The occipital lobe. Thank you. So somehow the signal has to make it from the retina all the way back to the occipital lobe, and the pathway is shown here. So you have the rods and the cones first, and they transmit the signal forward to bipolar cells, and then on forward to ganglion cells, and then you have other horizontal cells and amacrine cells, which help in visual activity as well. But the ganglion cells are what transmit the signal onto the optic nerve. Okay. Computer's a little slow. So you can see there the pigmented area, the rods and cones, and the bipolar cells, the ganglion cells, and their axons are what form the optic nerve and transmits the signal on back to the optic chiasm, the optic tract, and on to the occipital lobe. So here, if you were to look into a person's eye with an ophthalmoscope, you would see the blood vessels, and then you would also see this area, which is the blind spot, and the fovea there. In fact, I was just looking at the lab manual, and you all are going to do some of the exercises in 24. And there's that exercise that allows you to visualize, look at a speck, and then you'll see that one part of your visual field goes blank. So you'll see the blind spot. The fovea centralis. So as light enters the eye, whatever object you're looking at, the image is going to fall right there in the center of the fovea centralis. And aqueous, I mentioned aqueous humor, which is the fluid circulating within the eye. And intraocular pressure is the fluid pressure in the aqueous humor, and it helps to retain the shape of the eye. And if a person has glaucoma, the problem is their intraocular pressure is too high, and that starts to damage the retina. And the lens, so we kind of mentioned all of these. The lens fibers, cells in the interior of the lens are filled with crystallins, which provide clarity and focusing power to the lens. So remember, the lens is what allows us to focus, allows our eye to focus the image onto the retina. And cataracts, you all have heard of that, a condition in which the lens loses its transparency. So the lens becomes cloudy in cataracts. And that's one of those things that will happen to just about everyone if you get old enough. Light refraction, when you see refraction having to do with the eye, the eye has various refractive components which bend light. So refraction just means the bending of light. And so the bending of light by the cornea, so when light hits the cornea, you'll see that the light rays bend slightly. And the focal point is a specific point of intersection on the retina. So wherever the light ends up meeting up with the retina is the focal point. And the focal distance is the distance between the center of the lens and that focal point. First, let me show you the bending of the light rays. So you can see, if you just look at the bottom picture here for a moment, when depending on what you're looking at, even if you're not looking directly at something, all of the light is passing through the cornea, and it slightly bends. The cornea makes the light bend in kind of a converging fashion, and then when it hits the lens, it bends again. So those are refractive components of the eye, and that is what allows us to be able to make sure an image makes it to the proper part of the retina, which is the fovea centralis. Okay, there was something I skipped. I wanted to go back to accommodation, and that's one thing you need to know for the test, is how the lens changes shape in order to focus the image that we're looking at onto the retina. So accommodation is how our lens changes shape in order to focus an image on the retina. And astigmatism is a condition where light passing through the cornea and lens is not refracted properly. Okay, so the eyeball shape is not, the shape of the cornea, for example, is not perfectly spherical like a basketball. It's more shaped like a football. So when light rays come in, they don't bend the way they're supposed to. So the image doesn't fall on the correct part of the retina. In fact, it may have two focal points, and the visual image is distorted because of that. And what that means is, for example, if you were looking at this person's, the light coming in, instead of the light bending and landing on the fovea centralis, that would not be the focal point. There may be two focal points, so that when the image or when the signal gets to the brain, it's kind of confusing. So that's what happens with astigma. There ends up being more than one focal point. Okay, so with accommodation, so the eye or the lens responds differently if we're looking at something close to our face or looking at something far away. Okay, so for close vision, so if you're looking at your book, for example, this is what happens to your eye. So the ciliary muscles contract. Remember what those are? They're connected to the suspensory ligaments, and they control what? The shape of the lens. Okay, so what they do, if you're looking at something close to your face, those ciliary muscles contract, and that causes the lens to become more round. Okay, so if you look at the top picture, you'll see that the lens is more oval shaped, whereas the picture on the bottom, the lens is flatter. So that's a accommodation. So for distant vision, so when you're looking at me, since I'm far away from you, your ciliary muscles relax, and that causes the lens to flatten out. Okay? And all of that is to make sure that the image you are looking at falls on your fovea. So if the lens wasn't working, then the image would not fall properly on your fovea centralis, and the image would be distorted. All right. Image formation, so light from a point at the top of an object is focused on the lower retinal surface. And that should be obvious. If you look straight ahead, you're not looking at the ceiling, but you can see the ceiling, right? So the light rays from the ceiling are coming at an angle, so it's going to fall on the lower portion of your retina. And the floor, the light rays are coming up, so they're going to land on the upper portion of your retina. And therefore, whatever you're looking at is inverted by the time it makes it to your retina. Okay? And you all may have seen that before. So by the time the image, like I said, makes it to your retina, it's inverted. And what happens is the brain receives the information and just flips it right back over. They did an experiment, I remember reading about an experiment long ago, where they somehow took the eyeballs of a frog and made them turn them upside down. And somehow, I'm not sure how they knew what the image was that made it to the brain, but when they turned the eyeballs upside down, the frogs were actually seeing things upside down. Yeah. So if your eyeballs were flipped, you would see the room upside down. But then, after a few weeks, the brain fixes it. So after a while, they were able to adapt to that and see things the way they were supposed to. So somehow when the signal makes it to the brain kind of reorients things the way they should be. Mm-hmm.
I was reading in National Geographic, a man got into a crossfire, and he had a brain injury, and somehow it affected his vision, so he sees everything upside down.
Really?
Yeah.
Interesting. No, and it is interesting how the brain works. But I'm wondering if it over time, if it will fix itself. But really, it's the brain. Like I said, it's not the eye. So his eyes are okay. So some part of his brain, I guess the part that inverts the image, is not doing that. So that must be the explanation.
Distances and stuff. I mean, like, it's really hard.
Interesting.
They can't start doing surgery until they figure out what why he can see upside down.
Right. So he can see, but things are just upside down.
Well, they also talked about an article how when he turns his head this way, it automatically switches. Like he still sees upside down. Like it just, you know how when we see something and it's upside down, but we turn our head.
Right. So if he's upside down.
Like it turns.
Interesting. It is. Anything to do with the brain is pretty interesting. Now, so myopia. What is myopia? Are any of you myopic? Anybody wearing contacts or glasses? Yeah. So, myopia is nearsightedness. Any of you nearsighted? What does nearsighted mean? Nearsighted means you see things better when they're close to your face. And farsighted is when you see things better further away from you. And older people, if you have grandparents, sometimes you'll see them reading the paper pull it way back. So as we get older, we will become more farsighted. But what is actually happening there? What's wrong with the eyeball? So again, the eyeball is supposed to be perfectly spherical, like a basketball. So a person who's myopic, their eyeball is a little bit longer. Okay? And let me show you a comparison there. That wasn't a good one. So if you compare this eyeball to that eyeball, you can see the difference. So this one, the eyeball is too long, and the focal point doesn't end up falling on the retina. So the perfect focal point is here, but that spot is not hitting the retina. So when the light makes it to the retina, it's not the sharpest possible image. So a person who has myopia is giving corrective lenses that are diverging lenses, and that allows the focal point to fall actually right there on the center of the fovea centralis. So the eyeball is too long, the focal point ends up being before or in front of the retina. Okay? It's because the eyeball is too long. And compare that with hyperopia, which is farsightedness, and you can see the focal point ends up what? Behind the retina. So that's the problem. The perfect image is going to be at the focal point, but the focal point is not on the fovea centralis, it's slightly behind it. So the light that actually passes through the fovea is going to be slightly blurred. Okay, so hyperopia is farsightedness, and that happens if the eyeball is too shallow or the lens is too flat. So when we become older, we become farsighted because the lens loses its elasticity. And that's a form of hyperopia called presbyopia. Presbys means old man, and that's what happens to us when we become older. And so accommodation problems. Any of you had LASIK or know someone who's had LASIK surgery? Yeah, it is. So in this procedure, what they do is change the shape of the cornea. Hello. And this usually works well, so patients are able to achieve normal vision. And I hear it's really quick. I've thought about having it myself. All right, rods and cones. So the visual pigments. So remember the pigment layer of the retina, the derivatives of rhodopsin, opsin plus retinal. So these are the retinal is synthesized from vitamin A. So these are the pigments that actually activate the rods and cones, what the rods and cones respond to when light comes into the retina to that pigmented layer, the most posterior part of the retina. Color vision, integration of information from red, green, and blue cones. So we have cones. Remember, cones are for color vision, but we have 3 different types. And those are the colors of red, green, and blue that these cones detect. Inability to detect certain colors results in color blindness. So for this one, can everybody see the number in the center there? And you all probably have seen tests like this when you were younger, especially.
I saw it in a movie where military, when soldiers were entering, they would have to take the test, and if they couldn't tell what the number in the automatically found out their colorblind, they were sent.
Yeah. Yep, so if you can't see the 12, that probably means you're colorblind, and the problem is you don't have those red cones. Okay, and color blindness. Remember, it's a genetic disorder, but it's sex-linked, right? So remember, sex-linked disorders. What that means?
Yeah.
From Biology 103. What?
So women, we have X tags, so we could be carriers, but we have, because we have another X, it compensates for that X that is passed on, so it doesn't show. But because males have an XY, they don't have one to compensate for the other, so it shows.
Very good. And so, because of that, so if you're a female and you are colorblind, all of your sons are going to be colorblind. Oh, yeah. And because of what she just said. So if you have, remember you have 2 alleles for each trait. So if you're a female and you have 2 bad alleles, both of them are on the X chromosome. So your son is going to get one of those, right? And he's going to have an XY, but he doesn't have anything to mask it. He doesn't have a good chromosome to override the recessive one. So that's what happens with sex-linked. Depth perception obtained by comparing relative positions of objects between left and right eye. So what this is explaining is that when you look at an object with 2 eyes, you can judge how close that object is to you. But if you just have 1 eye, and you all can actually practice doing this, if you just have 1 eye, it's hard for you to tell how close something is to you. Okay? So you all may do that in lab. I think there's an exercise where you can do that. And basically what you do is first close both eyes, because when your eyes are open, you know where everything is. And when you open your eyes, just open 1 eye or cover 1 eye and try to judge whether you're close enough to touch someone. Okay? So if I just had 1 eye, I may feel like I'm close enough to you all to touch you. And when I was explaining this to one of my students a year or so ago, he told me he had a son who was about 16 who lost an eye playing with fireworks. And he said every now and then he'll mess with him by throwing him a tennis ball or something. And he said his son, he was really good, an athlete before the accident, but since the accident, he can't really tell when the ball is going to get there for him to catch it. And that's an example of loss of depth perception because you only have 1 eye. So that's, I guess, why we have 2 eyes instead of just 1. Okay. The anatomy of the ear. So you all know this stuff. So as far as the test, cochlea. So there's quite a bit more information you all can learn about the ear in the textbook. The organs, you definitely need to know the organs for hearing and equilibrium. The cochlea is the organ of hearing. No, for hearing. The cochlea is for hearing. The vestibule, the whole vestibular apparatus, is for equilibrium. So that's where the receptors are. And you all remember the vestibular cochlear nerve? It bifurcates in order to innervate the cochlea and vestibule.
So you have from the spiral canal, like, is it because of the shape of it?
Yeah. So it's also called the spiral organ. Okay. And I'll show you all a picture in a moment. So definitely know those 2, their functions. And the auditory tube is also called what? Auditory tube, eustachian tube. It's also called the eustachian tube. And that's important because it equalizes pressure on both sides of the eardrum. So if there's a buildup of pressure on one side, it causes the ears to pop. You ever been on an airplane and your ears start popping? Well, that's because there's a difference in pressure on the two sides of the eardrum. So the atmospheric pressure here is different from the pressure inside of the inner ear. Vestibule. The vestibule for equilibrium.
Is it vestibule or vestibular?
AR just means pertaining to. So if you see vestibular, that word means pertaining to the vestibule. Cochlear means pertaining to the cochlea. Okay, so how does sound work? How do we hear? So we know sound travels in waves, right?
Yeah.
And those waves hit our eardrum, and that starts the process. So the waves travel through the external acoustic meatus, bumps into the tympanic membrane, and the tympanic membrane vibrates. And what's connected to the tympanic membrane? The ossicles, right? So that vibrates the ossicles, which vibrates the oval window. And there, once the signal gets inside, I'll show you all that here in a moment. Hopefully I have a picture. And another thing, before I get to that, the inside of the cochlea, are these two muscles of the middle ear, the tensor tympani and stapedius muscles. And they're important because, you know, if you're exposed to very loud sounds, especially sudden, like a gunshot, if you ever go to the gun range, you put on earphones to protect the ear drums, because the ear drum can tear if the sound is too loud because it vibrates too much. And what these muscles do, they're supposed to prevent the vibration from becoming that drastic. But they only work if it's over a long period of time. So if it's a sudden loud sound, as I just said, that happens too fast in order for those muscles to respond. So they protect the eardrum from over vibrating due to loud sounds. And so it stiffens the tympanic membrane, reduces movement of the stapedius at the oval window. I hope I have a picture. So yeah, so you see here the inner ear. So in the inner ear, you have these spaces. That's why it's called the vestibule there, and in the cochlea as well. And you have something called a cochlear duct. And the signal coming in vibrates this kind of gel-like fluid that's in the ear. And when the signal makes it to, I'll come back to this in a second. I can't believe I don't have that. So I'm missing a couple of my slides. But what this one is showing you is equilibrium. So in the inner ear you have the vestibular apparatus. And you also have the semicircular ducts. And what they do is allow us to interpret the movement of our head, and also whether we're moving fast or slow. And there's material inside of those components of the ear called endolymph. Endolymph movement along the length of the duct stimulates the hair cells, and it's the stimulation of the hair cells that activates receptors. And you can kind of see it on this one. So this gel, we have the otolith, and whenever a head moves, this otolith is kind of heavy, so it moves the gel, and that moves these hair cells here. And when the hair cells moves, that's what's activating the receptor and transmitting the signal onto the brain. You all will have a much better picture of that in your textbook. So try to get as much understanding about that as possible. I usually don't ask too many questions.